Oops. Okay, so we're going to pick things back up here at the inheritance patterns. Now, Navy explained that glycophorin A and glycophorin B are the home to the antigens M and N, big S, little s, and U. These genes, the GYPA and GYPB genes, are located on chromosome 4, and they code for the alleles that produce the M and N antigens for glycophorin A gene, and the S, big S, little s, and U antigens on the glycophorin B gene. So it is important to remember that these antigens and the antibodies to these antigens demonstrate the dosage effect. So potential antibodies should only be ruled out when we have a double dose of these antigens on the red blood cells or they're homozygous. When we're doing our antibody screens and antibody panels, if we're trying to rule out a anti-big S, for example, we need to be sure that that cell only has the big S antigen and not the little s antigen. Because these genes can display codominance, meaning that the M and N or big S and little s antigens can be present on the same cell at the same time, when we're doing these screens and panels to rule out that antibody, we need to have a double dose or homozygous cell. Maybe also mentioned that there are 50 different antigens in the MNS blood group system. This happens because of the mutations that occur during mitosis. Whether that is a unequal crossover of the genes or an insertion or deletion, any one of those mutations can give rise to MNS blood group system. And that's why there are so many found in this blood group. So speaking of some unique antigens, let's talk about the UVAR and the ENA antigens. So the UVAR phenotype is found in people who are thought to be U negative, which is very rare. U is nearly ubiquitous, but they demonstrate a weak reaction to an anti-U antibody. Now, this variant of U has a mutation in the glycophorin B, so it does not express big S or little s, but it has certain epitopes of the U antigen. I thought that this was analogous to the mosaic form of the D antigen in the Rh blood group system. So ENA, on the other hand, is a very high incidence antigen. It happens nearly everywhere, and it refers to the portions of glycophorin A that are unrelated to the M or N antigen. So it's either in between or below these antigens on glycophorin A. So this antibody is a very serious antibody. It has been implicated in severe hemolytic transfusion reactions and hemolytic disease of the fetus and newborn. And because this is a very high incidence antigen, it would be very difficult to find a compatible blood unit for somebody who has formed the anti-ENA antibody. Now, ENA stands for envelope, and that's the name that it was given. Okay, talking about some disease associations, the MNS antigens serve as receptors or a binding site for complement, bacteria, and viruses. I thought this was an interesting fact about the MNS blood group system, that certain strains of the E. coli bacteria can use the M antigens as a receptor and binding site to enter urinary tract cells and cause infection. Plasmodium falciparum can also use both the glycophorin A and glycophorin B as a receptor and binding site in order to enter red blood cells. Now, this could cause a problem in malaria endemic areas for any person with these glycophorins, which is nearly everybody. All right, let's get into the specific antibodies. So anti-M is normally a cold reactive antibody and is significant only if it is reactive at 37 degrees Celsius. If we remember the high elemental P mnemonic, we have the M in there. However, one key difference here is other cold reactive antibodies are of the anti-M class. while here 50 to 80% of anti-M antibodies are going to be IgG or have an IgG component. Some of these anti-M antibodies react better at a lower pH. So they'll react best at a pH of 6.5 to be specific. So if we're trying to increase reactivity of the anti-M antibody, we can acidify the serum that we are using in order to enhance that reaction there. Other enhancement methods include increasing the serum to cell ratio, so we'll have more antibody to react with those epitopes on the cells. We can also increase incubation time to allow for sensitization to occur. And an interesting fact here is that anti-M antibodies might react with stored or reagent blood cells, but will not react with freshly collected red blood cells, and that is due to the potential pH difference there. So anti-M rarely causes hemolytic transfusion reactions or HDFN. It is a non-RBC immune antibody, meaning we don't need to have an exposure to this antigen to form an antibody. Anti-M does not bind complement, regardless of the IgM or IgG class of antibody. And an interesting thing here is that this antibody is more common in children than in adults. And that could be due to a number of reasons. Okay, moving on to anti-N. So this antibody is similar to anti-M. However, it is less common than the anti-M antibody. So this is a cold reactive IgM or IgG antibody, and it is not normally clinically significant. It doesn't react with enzyme-treated cells. The demons destroy Chadell-Rogers, right? They have the MNS blood group system in that. And this also demonstrates dosage. So something important to note here is that rare cases of anti-N have been reported to cause hemolytic disease of the fetus and newborn. So even though it is normally insignificant to transfusion medicine, it has the potential and the ability to cause serious reactions here. This is also a non-RBC immune antibody. Okay, moving to the S, big S, little s antibodies. These are mostly IgG antibodies, and they are reactive at 37 degrees Celsius, meaning that they are clinically significant antibodies. So these antibodies also demonstrate dosage, but not as strongly or avidly as an anti-M or an anti-N antibody. So with anti-S and big S and little s, these are serious antibodies when they are found. They have been recorded in very severe cases of hemolytic transfusion reactions and cases of hemolytic disease of the fetus and newborn because they are of the IgG subclass. The antigens on the red blood cell are close enough that if an antibody binds to those antigens, they could also bind complement, which would increase the severity of a reaction. Now, these are RBC immune antibodies, so we won't find these naturally occurring in people. We'll need to have an exposure to these antigens in order to form an antibody. Okay, anti-U. This is also a serious antibody. U is found in nearly every population. Only 1% of African Americans lack the U antigen. So it is a very, very high incidence antigen. Anti-U is normally IgG, and it is reactive at 37 degrees Celsius. So if it is found, it is serious and significant in our transfusion medicine. It has been reported to cause severe cases of hemolytic transfusion reactions and HDFN. However, this is very rare because the U antigen is in such high quantity that we will rarely find somebody who can actually form an anti-U antibody. Okay, and then moving on to our last slide here, the anti-NF antibody. So this is an interesting antibody in this blood group system. It is only seen in patients who are being treated for renal disease and are being treated with dialysis on machines that are cleaned with formaldehyde. Now, this anti-N antibody will react with any red blood cell that has been in contact with formaldehyde, whether it's a N positive or N negative cell, it will react with it. I thought that was interesting. This antibody does not react at 37 degrees Celsius and is therefore insignificant in transfusion medicine. Something to note here that the anti-NF antibody will disappear as soon as cells treated with formaldehyde are removed from the body and the formaldehyde exposure stops. So that is the MNS blood group system. Hope you enjoy and learned lots. Thank you very much.